All right, we're ready to move on. Okay, so I know there was still some more questions, and we didn't get a chance to go through all of them. So I'll hang around after the next lecture. If things go beyond that, we can walk out in the hall. Okay, and I mean I hate to make you miss part of your next lecture, but if you miss a couple minutes because you had a question, I think that'll be all right. All right, so our next topic, control of gene expression. So here's your learning objectives. So gene expression, so this is a very, very general term, so it refers to all the processes that result in the production of a functional gene product. Okay, so that means the transcription of an RNA molecule, the processing events that occur in the nucleus to that primary transcript, the transport out of the nucleus, all those things ultimately are affecting gene expression, including all the things that happen in translation, all the post-translational modifications that occur, those are all part of gene expression. Because ultimately what you want to end up with at the very end is a mature protein of some sort that's going to perform some function. So any event that can affect that is part of gene expression. But the primary level of the control of gene expression is going to be at the level of transcription. So whether or not and how much RNA we make, messenger RNA we make for a particular gene. So there are certainly other ways we can influence it, and we'll see a few of those, but the primary one is by controlling how much mRNA we make. And that happens in both prokaryotes and eukaryotes. Now, eukaryotes actually have additional control mechanisms, and we're going to talk about those because of all these processing events that occur. So all those processing events we talked about that were somewhat, at least in many cases, unique to eukaryotes, are part of gene expression that's unique to eukaryotes. So, long story short, eukaryotic gene expression is going to be a little more complicated than in bacteria. Now, we can classify genes into two groups. We've got our housekeeping genes, and then we've got our regulated genes. So our housekeeping genes are genes that are constitutively expressed. That means they're on all the time. Okay, and there are definitely genes that need to be on all the time. They have what is loosely termed housekeeping functions. These are things that have to occur in every cell all of the time to maintain life as we know it. Okay, basic cellular functions. And usually in biochemistry, you could actually make the argument that these are absolutely the most important things, because without them, the cell would be dead and you would be dead. But they're not so interesting because they're not differentially regulated. And in biochemistry, most of the time, we're really interested in the things that are differentially regulated. And we're also, in medicine, we're interested in health and disease. Well, if you mutated and rendered dysfunctional a housekeeping gene, you wouldn't be alive, so there would be no disease to have. So most of the diseases that we're going to talk about are less severe than that, and they're usually going to be inside some type of regulated gene. Or at least these are the most interesting ones from a very selfish human perspective. So our regulated genes are required only in certain cell types or only under certain conditions. And so we have different control mechanisms that are going to determine whether or not we turn those genes on or off. And that's what we're going to look at today. Okay, so first thing we're going to look at is the lac operon in prokaryotes. How many people have seen lac operon before? Is the lac operon fair game for MCAT? It is, okay, so you guys have already seen this. Awesome, man. You're going to have a lot of hundreds on the biochem portion of the exam. Can't wait. Really, I do expect you guys will do well. Not as hard as you think. All right, so let's look at the LAC operon. I guess you're already a little bit familiar with it, but we're going to look at it. Is there a question, or we just, okay. I saw somebody's picture coming up there. So this is a prokaryotic model. This is probably the most famous one, so that's probably why I think almost everybody's hand went up. And so this is going to affect some gene expression in bacteria, in this case in E. coli. So glucose is actually the preferred carbon source for E. coli. It's actually the preferred carbon source for many, many, many bacteria. Most things like to try to use glucose if it's available. However, E. coli can use other sugars, okay? However, if you're going to use those other sugars, you need to make enzymes that are going to process these other sugars, okay? In order to make those enzymes, that requires energy, okay? You already know how intensive it is to make a messenger RNA and to make protein. Well, unlike human metabolism and eukaryotic processes, bacteria tend to be very, very efficient. If you look at their chromosome, there's not much extra there, so they tend to be very, 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 very efficient. So they're only going to make these enzymes to process these other sugars when those other sugars are available. And in this case, also when glucose is absent. So if glucose is there, it's always going to use glucose. Doesn't matter what else is there. If glucose is absent, then it's going to try to use something else. But what it's going to try to use depends on what's available. Okay? And in this case, we're going to be talking about lactose. This is another alternative sugar. And so you actually have to, so the genes involved in metabolizing lactose. Now, with any alternative sugar, ultimately you can fit things into the pathway that processes glucose, which is glycolysis. We'll talk about it a little bit later on. But you'll have a few unique enzymes before you can get to that. And you're only going to produce those unique enzymes if that sugar is present and glucose is absent. So ultimately, those two things are going to determine whether or not the LAC operon is turned on. So operons are unique to bacteria. I think there is actually they found some eukaryotic operons, so they were thought to be unique to bacteria, but certainly they're much more common in bacteria. And so what happens, what an operon, the definition is basically you have a single sort of promoter region that actually controls the expression of multiple genes. In this case, we can make a polycistronic messenger RNA, which we had already talked about what that is. And the LAC operon, which controls the production of these enzymes involved in metabolizing lactose, only on if glucose is absent and lactose is actually there. So how does that actually work? Okay, so the first thing we got to look at is here's what's going on. Here's a basic look at the gene. I don't care about LAC-Z, LAC-Y, LAC-A. Just know these are genes involved in lactose metabolism. They're going to do something involved in lactose metabolism. Okay? So we need those if we want to process lactose. If we're not going to use lactose, we don't need those. Okay? This LAC-I gene, that actually encodes a repressor protein, which we're going to show you shortly what it is. This repressor protein, so this is on all the time. That LAC-I gene product is always present, okay? And it's actually the repressor we're going to see shortly. And it actually can bind to this operator sequence, as we're going to see. So we actually have 3 regulatory sequences that we're going to talk about that are on the DNA molecule. So these are in this promoter region that are going to determine whether or not we turn on this LAC operon. We synthesize the messenger RNA to produce these proteins. We do have our core promoter region, which we've already talked about. That's common to all promoters. That's there. Okay. Well, that doesn't really help us, determine whether we're going to turn that gene on or off. It's not anything special. Okay. But we have a couple other sites on the DNA. We have a CAP binding site. I'll show you in 1 second what that does. So some cap, as the name implies, cap can bind to it, but I'll show you what that is. And then we have an operator sequence. And the gene product of this LAC-I, the repressor protein, is actually what can bind to that operator sequence. Okay, so in order for transcription to occur, in order to produce these genes involved in lactose metabolism, we have to have two things that have to happen. Now, I told you, before we even look at the molecular details, I told you two general things that have to happen, right? Glucose has to be absent, and lactose has to be there, okay? Those two things, the absence of glucose and the presence of lactose, are the two different molecular pathways that contribute to whether or not the LAC operon is turned on. And in order for it to be turned on, we have to satisfy both those conditions. Glucose not there, lactose there. And so when glucose is there, what ends up happening is you have this cap-camp complex bound to the cap binding site. I'm going to get into the details of that in 1 second. And if lactose is around, the repressor will be removed from the operator. You satisfy these two conditions, you get transcription of that polycistronic mRNA, and then ultimately expression of those genes. So let's look at glucose first. Okay, so there's a couple terms I need to define here. CAP, catabolite gene activator protein. CAMP, or cyclic AMP. We've already talked about, I think we've already talked about cyclic AMP somewhere. Certainly you'll see cyclic AMP all over the place. It's a signaling molecule. Actually, we'll see cyclic AMP used again later on in this lecture for a eukaryotic signaling pathway. So it's used in both eukaryotes and prokaryotes for different, very common signaling molecule. So if cyclic AMP is produced in the bacterial cell, it will bind to cap and form this complex. This is the activated complex which can actually bind this cap binding site. Okay, so as we go through the LAC operon, one of the things I want you to be, and if you're trying to study this, this is how I would study it, okay? Write down on a piece of paper, when you think you're comfortable with it, write down on a piece of paper, glucose present, glucose absent, lactose present, lactose absent, and tell me what happens in each one of those. If you can do that, you know everything you ever need to know about the LAC operon, and you'll be able to figure out whether it's going to be turned on or off. So if glucose is present in this case, that actually inhibits this adenylyl cyclase. This is the enzyme that makes cyclic AMP. And if that cyclic AMP is not there, so it inhibits that enzyme so we don't make it, it doesn't bind to cap, and therefore it doesn't bind to this cap binding site. So what happens is you need the cap-camp complex bound there in order to efficiently initiate transcription. What I like to visualize in my head, my simple way of thinking about things, is I like to think of this needs to be there to kind of push that RNA polymerase forward. Okay, so that cap-camp complex needs to anchor there to kind of shove that RNA polymerase forward. Okay. Now, so if glucose is present, it's not going to be there. It's not going to give that RNA polymerase that shove it needs. Therefore, you're not going to get transcription of the genes. Now, if glucose is absent, that's going to activate this adenylyl cyclase enzyme, and it makes that cyclic AMP. That cyclic AMP binds to cap. Therefore, it goes to the DNA and sits on that promoter region, that cap binding site. Now you've got the appropriate binding to actually kind of shove that RNA polymerase forward, saying, get moving, make these genes so we can process this lactose. Okay? And so that's when glucose is absent. Okay? So you're going to have to meet two conditions, but there's one of the two conditions. Glucose is absent, that cap-camp complex is there to try to shove that RNA polymerase forward. Okay? That's one of the two conditions that needs to be met. The other condition involves this repressor and the operator. So this repressor is encoded by the LAC-I gene. It's always going to be present. It's always going to be there. And it can bind to the operator sequence. So the RNA polymerase, remember, is going to be somewhere right here where this core promoter region is. And I talked about whether or not you had that complex to kind of shove it forward. Well, if the repressor is bound to this operator sequence, it's actually going to physically block the RNA polymerase from moving forward. So, for example, even if that CAP-CAMP complex is there trying to shove it forward, if the repressor is there, it's not going to move. Okay? And so when is the repressor bound to the operator and when is it not bound to the operator? Well, if lactose is present, okay, a small amount of allolactose will actually be produced. And so that's a non-enzymatic pathway. So it's a spontaneous conversion. You get a small amount of lactose produced, and this is actually what can bind to the repressor. If you get this allolactose bound to the repressor, it no longer binds to the operator. So that repressor protein is going to bind to one or the other. It's either going to bind to the operator or it's going to bind to allolactose. It doesn't bind to both of them. So when it's bound to allolactose, it's away from the operator sequence. Now that RNA polymerase can actually move forward. Okay? So when lactose is absent, that repressor is going to be bound to the operator. That's going to physically block the RNA polymerase. When lactose is present, allolactose is going to bind to that repressor. Therefore, it's no longer going to be bound to the operator sequence. Therefore, you've removed that physical block, and now that RNA polymerase can move forward. Okay? So, ultimately, in order to turn on the lac operon, which is what's going to happen in B here. By the way, if you use this figure to study from, one thing that really annoys me about this particular figure is really they shouldn't have shown in this one right here. They shouldn't have shown that operator sequence there. That's an error that they haven't. This is the figure from the 6th edition, so they haven't. I don't know why they put that there. It shouldn't be there. Okay, that makes this figure very confusing if you're trying to figure out why that's there. Because if lactose is there, it should be bound to the operator, right? So the operator shouldn't be there. So if you use this to study from, just don't let that confuse you. So that should not be there. And actually, yeah, no, that's right. Okay, so when are we going to actually turn this lactose operon on, this lac operon on? Okay, we have to meet two conditions, right? I told you glucose has to be absent, lactose has to be there. So what happens if glucose is absent? Glucose is absent, we activate that enzyme, adenylyl cyclase, right? Cyclic AMP builds up, binds to CAP. That cyclic AMP CAP complex will actually bind to this CAP site, and it wants to shove that RNA polymerase forward. Okay, that's one of the two requirements that needs to be met. And if lactose is there, allolactose will be made. It will bind to that repressor protein, and it will be removed from the operator. So now you can actually shove that RNA polymerase forward. So both of those conditions have to be met in order to get efficient transcription. That repressor needs to be removed to get out of the way, and this CAP-CAMP complex needs to be there to kind of shove the RNA polymerase forward. Okay? If either one of those two conditions is there, if the repressor is not there, or I'm sorry, let's say if the repressor is there, and they don't have an example there, but if the repressor is there, and the CAP complex is also there, you still won't get transcription, okay? Because the repressor will actually block it from occurring. If you get that CAP-CAMP complex bound there, oh, I just said that, right? And the repressor's in the way. So if you remove the repressor, but you don't have the CAP-CAMP complex there to kind of shove it forward, it still won't get active transcription, okay? So once again, I think when you're studying this, there's a couple different ways to approach it. You can recognize, well, what from a very basic level is what has to happen? Well, we have to have an absence of glucose and a presence of lactose. Ultimately, how does that affect things? Well, in order to, if you have an absence of lactose, or I'm sorry, if you have an absence of glucose, this first thing is happening, the CAP-CAMP complex. If you have the presence of lactose, the second thing is happening. And once another easy or good way, I shouldn't say easy, a good way of studying this is right at the end, if you can write out on a piece of paper, what happens if glucose is there, what happens if glucose is not there? What happens if lactose is there, what happens if lactose is not there? If you know those four things, you know how this works. And then you can also figure out from those four things, what are the two things that have to happen in order to turn that lac operon on. Okay, so now we're going to move on to eukaryotic gene expression. And I made this little table for you. It's actually an adaptation of a table. I believe they have a similar table in the COMLEX review book, or it's actually USMLE and COMLEX review book. But I modified it a little bit, updated it a little bit. So eukaryotic gene expression tends to be much more complicated than the prokaryotic model. You might think the lac operon is pretty complicated, but it's really not. And hopefully by the time you're done going through it, it's really not too bad. Okay? If you're struggling with it, come see me. But all these different things are examples of things that can affect eukaryotic gene expression and do affect it. Modifying chromatin, DNA methylation, histone acetylation or deacetylation, things we've seen before. Enormous amounts of different specific transcription factors. Okay, those are things you may have seen before. We'll look at a couple of them today, real big ones. mRNA processing events that can affect both transcription and translation. The rate of translation can actually be affected. So, there are specific sequences that can make translation go faster or slower, or inactivate or activate translation. We're not going to look at any specific ones today, but they do exist. You can modify proteins, so that is an example of affecting gene expression, making an inactive precursor, which we'll see. Proteins can be turned over more or less rapidly, so the degradation rate is a way of influencing gene expression. One thing to remember about gene expression, in order for gene expression to truly work, to be able to regulate things, things have to be turned over. That means both messenger RNAs and proteins. They have to be degraded. Because if you didn't do that, then once you turn something on, it was on forever, right? So, and that's a key component of gene expression that people sometimes kind of forget about. So mRNA turnover and protein turnover are absolutely vital component and can very much influence gene expression and can cause diseases. And mRNA stability. We have diseases caused by mRNA being unstable, but you can influence mRNA stability as well by different processing events. You'll see one today. So the control of gene expression tends to be a little bit more complex. There's more things involved. Now, since the default state of chromosomal DNA could be considered off, so remember, DNA exists bound with histone proteins, and you have all these higher level orders of structure. And that's kind of the default mode for a DNA molecule in a eukaryote. Basically, eukaryotic genes, unless you turn them on, they're already off. Now, there are exceptions. There are things which can turn genes off, but they aren't as common as with bacteria. You don't see these repressors. So usually when we're talking about transcription factors and influencing gene expression, and we're looking at transcription, you're usually talking about something that's turning on transcription, because by default it's off. So just removal of those things turns transcription of the gene off. Now there are some exceptions, but 90% of the time that's what we're talking about. So we do have consensus sequences within the promoter. We already talked about those. We talked about the cat box and the top box. We didn't talk about that GEC box. That's just another core promoter region. Those are present in virtually all genes. But beyond those consensus sequences in the promoter, we often have enhancers. So These are regions of DNA that interact with specific activator proteins, or these transcription factors. Now, for some reason, and I'm not sure the exact logic behind it, but they started using this cis-acting regulatory elements instead of using this name enhancer regions. Enhancer is much easier to say, so I'm still going to say enhancer regions. But if you're using your textbook, that's these cis-acting regulatory elements. These are elements in the DNA that are going to enhance transcription. Okay, they're going to actually cause transcription to increase. Ultimately, if you increase transcription, you increase expression of a particular gene. So what are some of the features of these enhancer regions? These are regions of DNA that are going to influence gene expression, really enhance gene expression, make it go, make more of the messenger RNA. They can be close to the gene they're controlling, okay, and that's pretty logical, okay, they can be right near the promoter region, or they could be thousands of base pairs away. Okay, now that's weird. Why would they be thousands of base pairs away? There's a lot of weird things in the human genome and eukaryotic genomes in general. They tend to be enormous for some weird reason. And so these enhancer regions aren't necessarily right in that, what we would normally consider that promoter region. They could be thousands of base pairs away. They could be located upstream or downstream. That's weird too. They could be downstream. There's actually even some introns that have enhancer activity. So very odd. So how does that actually work? Well, it's, DNA can actually, move around, and so these enhancer regions can actually be brought close to the core promoter just by folding. Occasionally, you could even have an enhancer that's on a different chromosome. Okay, so these enhancers are odd. I don't think 20 years ago people would have predicted some of these things. They can act in a tissue-specific manner if the proteins, these DNA-binding proteins, are only produced in that tissue. And we'll see an example of some today. We're going to look at some genes that would only be, or a gene that would only be expressed to any significant degree in hepatocytes in the liver. Okay, and once again, you can bring the DNA back close to that promoter region just by folding. And here's a really simple example of folding, but certainly you can have DNA from way far away fold back and influence the beginning of transcription. So you also have transcription factors, and in the new text they call it transacting molecules. Transcription factors, that name has been used for so long, that's just what I call them, transcription factors. Probably what you've seen before as well, but your text has another name. So transcription factors are going to have a DNA binding domain, so they're going to bind possibly the promoter region or enhancer regions, but they're going to bind to DNA. They're also going to have an activation domain. Okay, so they're going to influence gene expression somehow. So they can either bind other transcription factors, they can interact with RNA polymerase II, which is shown right here, to help initiation of transcription, stabilize that complex. They could possibly recruit chromatin modifying proteins, so they could actually, if the transcription factor binds, it could bring in proteins that are going to acetylate histones and make that beads on the string model, so we can actually have active transcription. So these are all possibilities that could happen with transcription factors. And that's as detailed as we're going to get about it. Now you look at that figure and you'll be like, okay, ooh, there's a lot going on there, but don't make it any more complicated than it has to be. Okay, so I just want you to know the definitions as I did that. We are going to talk about a couple of specific transcription factors, and the only reason that I'm going to talk about those is because they are high yield. It is high yield information. You'll absolutely see this in your first aid study guide when you go to use that and things. Okay, So not all transcription factors are high yield. Actually, 99.99% of them aren't high yield. But here's a couple good examples of influencing gene expression in eukaryotes. There's this process, gluconeogenesis. I know you haven't seen that yet, but we will be talking about it shortly. This is making a new glucose molecule. And later on, I'll just give you some basics about why we would want to do this. So if we're in the well-fed state, that means you just ate. Insulin is going to be dominant. If you didn't know that, you will by the time we get through metabolism. That means you have plenty of glucose around. But later on, when you don't eat, like for example, when you're sleeping, you're going to run out of glucose. And eventually you're going to have to make glucose to replenish blood glucose. You absolutely have to have glucose in your blood in order to live. There are certain cell types and tissues that have a reliance on glucose, okay? And you have enzymes that are going to be involved in synthesizing glucose, okay? Those enzymes can be turned on or off or be at higher or lower levels, okay? And part of that's going to depend on your diet, but also part of it's going to depend on what state you're in. If you're in a state where you need to be making glucose, you're going to turn on the genes involved in that. One of those genes happens to be this PEB carboxykinase, phosphoenolpyruvate carboxykinase. One of the genes unique to gluconeogenesis. That's all I want you to know right now. If we're turning this gene on, we're trying to turn on the process because we need glucose for one reason or another. Now, whether or not that gene gets turned on is under the control of two hormones. Glucagon, okay, if glucagon's there, that signals that blood sugar is low. It means you're getting hypoglycemic. And the other hormone that can control it is cortisol. Cortisol is produced when you are stressed or when you reach starvation. Starvation is a metabolic term in biochemistry. When you get past the fasting state, and let's say it's about 12 to 24 hours when you start entering starvation. Okay, your body actually metabolically will react differently. But these two processes actually undergo a little bit different pathway in order to influence gene expression. Now, ultimately, both of these can turn on the gene. You can have one turning on the gene, the other turning on the gene, or both turning the gene on even more. So they can be additive as well. But the process of the signal transduction is a little bit different because glucagon and cortisol are very different hormones. They act in very different manners. That's because glucagon is water soluble and cortisol is not water soluble. It's fat soluble, it's not water soluble. So what happens with glucagon? You have a receptor on the cell surface, outside of the cell. So glucagon never actually enters the cell in this case. Binds to a receptor, and you have a signal transduction event that's going to occur. In this case, it actually uses cyclic AMP. So eukaryotes also use cyclic AMP as a signaling molecule. They also have a eukaryotic version of adenylyl cyclase. So the hormone binds to the receptor, turns on this adenylyl cyclase, and cyclic AMP is produced. This cyclic AMP will activate protein kinase A. It's not shown in this figure, but it happens at some point during this yellow arrow. I've got a figure in a couple slides here. I'll show you where the protein kinase A is shown. This activates CREB, ultimately. Protein kinase A, it's a kinase. Have you heard of a kinase yet? So kinase is something that's going to phosphorylate something. CREB is actually going to be activated when it's phosphorylated, okay? And CREB is a transcription factor, okay? It's not active until it gets phosphorylated. Once it's activated, though, and it's phosphorylated, it will go into the nucleus and bind to the enhancer region that it needs to bind to. The enhancer region, so CREB actually stands for the cyclic AMP response element binding protein. I'm just going to call it CREB. But it's got a corresponding element on the DNA, this cyclic AMP response element, that it binds to. And when it binds to that enhancer region, so now we have a transcription factor binding to an enhancer region, it's going to turn on transcription of that gene. In this case, the PEP carboxykinase gene is being turned on. We're going to produce that enzyme, so that way we can perform gluconeogenesis. And remember, glucose or glucagon was there because it was signaling blood glucose was low. We need to turn that enzyme. So it's one example. And then cortisol is another example. Also is going to turn on PEP carboxykinase gene, although through a different mechanism. So cortisol diffuses into the hepatocyte. If I didn't mention it earlier, this gene is going to be expressed exclusively in liver cells, a couple other cells too, but gluconeogenesis is a process that's unique mainly to the liver. See it a little bit in the kidney as well, but certainly it's not done in all cell types. So the cortisol in this case will actually diffuse into the hepatocyte. Cortisol is not water soluble, so it's fat soluble. Because it's fat soluble, it can actually cross membranes. So you'll see that with all your fat soluble hormones. They cross membranes. Okay? There is actually a receptor inside the cell as opposed to the cell surface in this case. So it binds to that receptor, and that cortisol bound to the receptor, in this case, it actually causes this receptor molecule to dimerize, and it will actually enter the nucleus and bind to a GRE. This is a glucocorticoid response element, okay? And it will turn on, once again, that PEP carboxykinase gene. But this is a very different mechanism, okay? So you had that water-soluble hormone that never entered the cell, that had that signal transduction cascade, ultimately phosphorylated a transcription factor and turned on this gene. Here we actually have a hormone that crosses the membrane, interacts with a intracellular receptor, and in this case, the intracellular receptor and the cortisol itself bound to each other is the transcription factor. Okay, and that actually binds to the enhancer region and turns on the gene expression. So here we have a summary figure that shows, once again, cortisol can turn on this gene, glucagon can turn on this gene, or both of them can turn on the gene and get even more expression than either one of them alone. Okay? But anyway, cortisol dissolves or goes across the membrane, intracellular receptor binds to that GRE and turns on gene expression. Glucagon has a surface receptor, produces cyclic AMP, activates protein kinase A. This is the signal transduction part of it. Phosphorylates that transcription factor, which is going to bind to its enhancer element. Once again, both. So you don't have to have both of these events occur in order to turn on the gene in this case. Either one of them will turn on the gene. Both of them will turn on the gene even more. So if you have both glucagon and cortisol present, you're going to express even more of this gene. Because remember, by default, it's off already. So that's a little different than our other model. Okay, so a few more things. Didn't skip a slide, did I? Did. Okay. So a few more things about eukaryotic gene expression. One thing that can influence eukaryotic gene expression is alternative splicing. Okay, so many years ago, and most of you, I guess, most of you guys are in your early 20s, right? So used to be, when I first started teaching it, it was happening. So now it was when you guys were children. But when they first sequenced the human genome, there was a bunch of scientists who were involved in that process. It wasn't one person who did it. You know, it was a collaborative effort. They actually had a sign-up sheet, and they had a wager where all these famous scientists were trying to predict how many genes were going to be in the human genome, and none of them predicted that the number was actually going to be as low as it was. They were all incorrect. Every one of them guessed much more than there ended up being. Usually they were guessing in the 100,000 region. I think the lowest guess was somewhere in the 50,000 region. But it turns out that there's actually about 25,000 genes in humans. That was a real big surprise, because we'd like to think, we're pretty complicated, right? We're the most advanced organisms. We've got to have a lot of genes. Turns out we don't have nearly as many as we thought. And actually, there's organisms with much bigger genomes and organisms with many more genes in there. So how do we resolve how complex we appear to be with such a few amount of genes? 25,000 doesn't really sound like that many. At least to a molecular biologist, it doesn't. Maybe to you guys that sounds intimidating. I promise you we won't learn 25,000, but it's not very many. Well, one of the ways we can actually generate a diversity of gene products from only having 25,000 genes is by alternative splicing. And I wrote here, you certainly don't have to try to memorize this number 60%, because it jumps around all the time. I think now that number is actually much, much higher. I've seen some sources say they think it's 95% of genes undergo alternative splicing. So you remember those eukaryotic genes have those intron regions that get cleaved out. Because of that process, we can actually join different exons together, and therefore generate a diversity of proteins from a single gene. Okay. This also, this combined with things like enhancers that could be located all over the place, really kind of made us question what our definition of a gene was. And so, I've even got people coming up after class asking me about that. The definition of a gene is much more fluid now, because it used to be you just think of your promoter and what you're making, that's the gene. Now it turns out there's all these other things. If you're making multiple proteins from a single gene, is each one of those a different gene? I don't know. So it starts to become a little confusing. That's why it's very hard now to define exactly what do you mean by a gene. We use the term all the time in biochemistry, but it's a little bit of a fluid definition. One example, tropomyosin. This is something Dr. Dan Leishman may have talked to you about, an actin filament binding protein. This is one famous example where you get different alternative splicing events to produce different proteins with slightly different functions. Okay, so by joining different exons together, in some cases it performs a similar function, in some cases it's pretty different. Okay, tropomyosin is a good example. Another example of influencing gene expression is RNA interference, RNAi. This is actually relatively recent. You guys probably, because you're so young, have already all seen this. But even as of, actually, I think this only made the latest version of the textbook, the 6th edition. I don't think it was in the 5th, and it definitely wasn't in the 4th edition of the textbook. When I started, we were on the 3rd edition of the textbook we're using. So RNA interference. So this is a mechanism of reducing gene expression. So in this case, something that's going to reduce gene expression, but we're not talking about it at the transcriptional level, okay? By either repressing translation, so it can actually physically block translation, or more commonly actually increasing the degradation of specific messenger RNAs. We now know, and this was just, once again, this was discovered relatively recently, but this is a huge area of research still, and we're finding out all these fundamental roles for this process: cell proliferation, differentiation, apoptosis. It's probably involved in normal day-to-day metabolism in every cell, but it definitely has very fundamental roles in those. But I'm just going to give you the basics of how it works. So in this case, we have a very small messenger, or I'm sorry, a small RNA, a microRNA, miRNA. When I say small, it's about on the order of 20 nucleotides. So that's tiny for nucleic acid. And it acts as a guide strand. So basically, you have this mRNA in the cytoplasm that's going to be translated. If for some reason we want to, some signal comes in, okay, we need to shut this gene off. You can shut it off at the transcriptional level, right? But you're also going to have, for a period of time, you're going to have those messenger RNAs around that are going to continue to make proteins. If you want to shut those off, you have to have another mechanism. And what we have here is, what they'll do is you synthesize a small fragment of RNA that's going to be a complement to some region on that messenger RNA. We're going to use our same Watson-Crick base pairing rules and bind to that RNA, and that will be degraded. Double-stranded RNA is not normal, and a cell will turn over double-stranded RNA. Okay? One example of double-stranded RNA is viruses. So your cell has all these mechanisms. When it sees double-stranded RNA, it's either a double-stranded RNA virus or it's a virus that's single-stranded that's replicating. We need to attack that. So it's already got these mechanisms to attack and degrade RNA. And so this will actually reduce gene expression by turning over that messenger RNA. Okay, so your cells can make this particular or a particular small molecule to actually turn it over. Where this really became super popular amongst biochemists and molecular biologists is you can actually take advantage of this, make your own microRNA, which we call an siRNA, and turn off whatever gene you happen to be interested in a cell. It's really valuable because, first of all, this is an easy way of turning off a gene. The classic way of knocking out a gene in the genome and doing all that, that is very laborious, very time intensive. You're talking about weeks to months to knock a gene out. I could order this microRNA and have it in my hand tomorrow and do an experiment with it tomorrow. So that's how quickly you can do this. And also the other cool thing is you can knock out genes that you couldn't knock out the traditional way. Some genes are absolutely vital, so you can't knock them out. If we use this microRNA and we only put them in at a specific time, we can grow up the cells, add this microRNA and shut that gene off at a specific time and then see what happens, where we couldn't do that before. There were many genes we just literally could not knock out. So this has been absolutely invaluable to molecular biology in discovering the function of genes. Another high-yield example of something that could potentially happen, messenger RNA editing. Something we haven't looked at yet. This is a post-transcriptional modification in which we actually are going to modify a base in a mRNA molecule. We're actually going to change it to something else. You might be asking, well, why would you even want to do that? Is this a repair mechanism? No, I'm not talking about that. If you get some error from RNA polymerase, that RNA just gets turned over. This is a specific gene expression mechanism. So an example of where this is actually used, we have this protein, ApoB100. There's a truncated version of this protein, ApoB48. Now later on you're going to learn about these two proteins and what they do and their functions. The full-length function, or the full-length protein, ApoB100, has an additional function. So it's got more function than ApoB48. So ApoB48 is the truncated version. Now, I don't really have time to explain to you what those two proteins do, but just know that ApoB100 basically has two functions. ApoB48 only has one function. And in some cells, you want both functions. In the other cell, you only want the one function. In some cases, and the more commonly, we would do some alternative splicing. But in this case, we actually go ahead and edit the mRNA. And what we do is we actually change, and we want to make a truncated version in the cells that we want to make a truncated version. We're actually going to change a glutamine codon to a stop codon. It's very similar to a nonsense mutation. We're actually sort of, in a way, introducing a nonsense mutation. In this case, this is the mRNA, so it's not a permanent mutation in the DNA, but it's a similar effect. We've now produced an early stop codon. So from that messenger RNA now, we're going to produce a protein that's smaller. It actually happens to be about half the size. It's actually 48% of the size. That's where they got the name from. So, and these are two gigantic proteins that your guys are going to spend some time learning all about the function. But that's how ApoB48 gets produced from the same exact gene as ApoB100. Okay? And so I have the last thing, and these are things we've already talked about acetylating or deacetylating histone protein, so I'm just adding that in there with the explanation. And I already sort of mentioned this, but I'll just mention it one more time. Methylation, if you want a gene turned off, it's going to be highly methylated, okay? If you want a gene that's supposed to be more active, it's going to be much less methylated or hypomethylated. So methylation itself turns genes off. Very common eukaryotic gene expression mechanism. All right, so I'm going to hang around and answer some of your all's questions. And once again, if we go a little bit long, we'll take it out on the hall over there.